Hello, my name is Andrew Wilkie. I have a dual role because I'm a medical doctor, so I'm an honorary consultant in clinical genetics, but also as a professor of pathology in Oxford. I run a lab at the MRC Weatherall Institute of Molecular Medicine. My group works on a childhood condition called craniosynostosis, which affects the normal shape of the skull. So when a child's skull is growing, there are narrow gaps between the bones called sutures. And if one of these fuses early, then growth of the skull becomes distorted. And this in turn can affect the growth of the underlying brain. So this is a problem that I've been working on since I started the lab in the WIM back in 1993, nearly 30 years ago. So at that time, when I first started, there wasn't a single patient in whom we could pinpoint a particular alteration in a specific gene. But thanks to the incredible technological revolution that we've had over this period, with the ability now to sequence an entire genome of an individual, we can now identify a cause in just over a quarter of all patients with craniosynostosis. And I'm pleased to say that because of the amazing team that I've had working with me, many of the new genetic discoveries have actually been made in Oxford, pinpointing the causes of, for example, Apert syndrome and many others associated with craniosynostosis.
Hi, my name's Steve Twigg. I've been in the clinical genetics group for about 26 years now. I'm a basic scientist. I've spent most of my time as a geneticist, I think, identifying new causes, new genes that underlie craniosynostosis. But more recently, I've been doing a lot more sort of developmental biology.
Hello, my name is Tecmara Corona. I'm postdoctoral researcher in Professor Andrew Wilkie group, and I'm working on craniosynostosis. I'm using various model systems to elucidate the molecular basis for this disease.
My name is Edu. I'm a postdoctoral scientist working in the Professor Andrew Wilkie's lab.
My name is Hannah, and I work in a group as a researcher for the past 5 years. My main research interests are using microscopy to study huge data we are generating.
Hi, my name is Becky, and I'm a PhD student working in Professor Andrew Wilkie's lab. I'm researching genetic causes of craniosynostosis, but specifically looking at small changes in the DNA sequence.
Hello, I'm Isaac. I'm a DPhil student in the lab of Professor Andrew Wilkie.
Hi, I am Young. I'm also a DPhil student at Professor Andrew Wilkie's group. I'm also working on the genetics of craniosynostosis, but my project focuses on the large chromosomal abnormalities, which are called structural variants.
Children usually need at least one major operation during their first 2 years of life in order to correct the skull deformity. And because the surgery for this is very specialised, there are only 4 centres in England that are licensed by the NHS to perform this surgery. And one of these is based at the John Radcliffe Hospital in Oxford. So this is a great place to be based if you want to study craniosynostosis and its causes. So provided that the parents give permission, at the time that the child has their operation, we collect some blood for making DNA and further genetic studies.
One of the projects I'm working on in the lab involves studying a gene called ZIC1. This is a gene where we've observed multiple variants in patients in the NHS, but it's confusing because it results in craniosynostosis, but also lots of other features that don't typically occur with it. In our work, we take samples from patients in hospitals across the UK, take the samples back to the Wilkie lab, and then we can immortalise these cells and study them in greater detail to try and properly understand how these specific variants contribute in a very nuanced way to the etiology of craniosynostosis.
Craniosynostosis is enormously variable in its effects and also in its causes. So sometimes we think that events in the pregnancy, for example, the baby's head getting squashed inside the womb, or mum taking certain medications, may be responsible. In other cases, there's evidence that an alteration in the genetic program, that's in the DNA, causes the sutures to be sticky and to fuse prematurely. And then in other cases, despite a lot of research, we still can't be sure what's the cause in an individual child.
So to really understand what causes craniosynostosis, you really need to know what makes up a suture. So the only way to do that is to look at model systems and to identify each of the cell types that make up the suture, but then also to place those different populations within the suture. By doing this, this gives us a way into understanding what goes wrong when sutures fuse prematurely, as they do in craniosynostosis. It also helps us to understand how sutures develop normally. And by putting this information together, this will help us in future to design therapies, potentially to prevent refusion of sutures that sometimes occurs after operations.
So two of the questions that parents frequently ask in the clinic are, why did this happen to my baby? And could it happen again if I have another child? And to answer those questions, you have to be able to determine a precise cause for the condition.
So part of my project involves looking at the data generated from the UK 100,000 Genome Project, and this was a UK-wide initiative launched in 2012, which aims to do whole genome sequencing on 100,000 patients from across the UK with either rare disease or cancer. So as part of that rare disease grouping, some of them were enrolled with craniosynostosis, and so we can therefore access this data online and use that as a way to investigate the genetic causes of craniosynostosis, whether that be big changes or small changes in the DNA sequence.
I mainly use long-range genomic technology such as the nanopore sequencing and the bio-nano optical mapping. And bioinformatically, I also look at the structural variants within the 100,000 Genome Project.
I'm applying CRISPR-Cas9 genome engineering to look at the rearrangements and modifications which we actually see in real patients in the model system.
So in our work to identify what cell types are within the suture, we've focused on the coronal suture, which is the suture that runs from ear to ear. And we've identified we've shown that it's really much more complicated than we'd previously anticipated. But we've identified some really interesting cell populations within the embryonic suture, including a population that looks like a ligament. It has ligament-like properties which sits across the suture. Population underneath the suture which looks like cartilage, which is interesting because we know that cartilage is around before normal and abnormal fusion. And we've also identified progenitor population. So these would be stem cells, which give rise to lots of different cell types within the suture.
But we're not at the end of the journey. There's still new genetic causes to be discovered. And we now have the privilege of being able to look at the genomes of individuals sequenced by the NHS under the new testing programme, that is, provided the families have given research consent. And as well as discovering new genes this way, we can also peer into what I'd call the dark matter of the genome, because there's a lot that we still don't understand about the genome. It's a bit chaotic, but we suspect there may be some rearrangements of the DNA that in some cases might cause craniosynostosis.